back up one slide as we start renewing our information on that carbon fragment processing. Pyruvate is produced when we're talking about glucose. Glycolysis occurs in the cytoplasm. It breaks the glucose into two pyruvate molecules. There are several steps involved in that, breaking a 6-carbon glucose into two 3-carbon pyruvates or pyruvic acids, and processing them with a net yield of 2 ATP and 2 NADH. So we're transferring energy during that breakdown, and our profit is 2 ATP and 2 NADH, which are just carrying the energy somewhere else. This is depending on a mitochondrion that's a double membrane system and has 2 spaces, a smooth outer membrane that encloses the entire organelle, and in addition, a inner membrane that is convoluted. It's folded back and forth, producing an inner space and an outer space and a huge amount of membrane area. This is, again, a typical illustration of folding of membranes. The idea with folding membranes is to produce more surface area. And that more surface area is useful for any transfer of molecules across the membrane. The more area you have, the faster you can absorb or expel something. The second use, and more important here, is that all of these systems, as we'll see today, involve multiple steps. That a reactant is converted in the presence of the appropriate membrane to a product, or it would be better to say a reactant goes to intermediate 1 as a result of enzyme 1. The intermediate 1 goes to intermediate 2 as a result of enzyme 2, and these multiple steps eventually deliver a product. The fact that you have so much intermembrane space means you can take those enzymes and arrange them into enzyme complexes. So enzyme 1, 2, 3, 4, 5, 6 can be lined up on the membrane, so when the reactant arrives, it goes straight through the product. It doesn't have to search for that next enzyme. So, let me get arranged here. We've just generated this pyruvic acid. The pyruvic acid is shown here, pyruvate. That's in the cytoplasm. We're going to transport that into the mitochondrion and begin the citric acid cycle. Now, this is an older slide. And this is a core central chemical process in living cells that use oxygen. Not just ours, but all oxygen-using organisms have this cycle, but its name produces some confusion. Eventually, we're going to have to resolve that. The citric acid cycle results from the fact that citric acid is the first formed intermediate. See here as a 6-carbon citrate. It's now, in more modern terms, we more often call it the tricarboxylic acid cycle, or TCA, and its discoverer, Krebs, is sometimes credited by calling it the Krebs cycle. It's those 3 names, TCA, citric acid, and Krebs, are all for this same cycle. So pyruvate comes in and immediately splits off a CO2. So that bond energy is captured by this NAD to NADH conversion. It requires coenzyme A as a carrier. Now, the remaining acetyl group, and by the way, I want to point out, acetyl is an important kind of common point here. An acetyl group is a 2-carbon group that can enter into the citric acid cycle. And the reason I'm going to emphasize this, coenzyme A is basically the carrier that locks it up and then puts it in an energy form where it can convert that 4-carbon carrier to a 6-carbon carrier. So here's the 2 carbons being added. The reason I'm going to focus a little on this acetyl-CoA is it really represents the common step for carbohydrates, for fats in the form typically of triglycerides, and in the form of amino acids. The way these three act as nutrients is all by feeding acetyl groups into the TCA. We will return to this in just a moment. So back to the pyruvate to acetyl-CoA, and you contribute it to the citrate or 6-carbon beginning step. So when you add these two carbon, hydrogen, oxygen groups of the acetyl group, you're elevating the energy level of that 4-carbon beginning and to the citrate level. Now look what happens in the first two steps. You kick off a CO2 and go to 5 carbons. You kick off a CO2 and go to 4 carbons. So right here, by the way, all the intermediates are not drawn in here. All that's drawn are the CO2 delivery steps, and that happens early. Now notice something. Pyruvate's feeding in CO2, feeding in carbon. 3 carbons are being fed in, and 1, 2, 3 are kicked out. This is where the CO2 is generated, is in the approach to the citric acid cycle and in the first 2 steps. Now, there is enough energy released that using hydrogen in the matrix that surrounds these membrane systems, we make 1, 2 NADHs on this front end of the citric acid cycle. But we're now down here to a 4-carbon intermediate, but it is still more energetic than this 4-carbon over here. What we're going to do now is we're going to extract some of that energy. So what happens down here at about the 4 and 5 o'clock region of this circle is that actually it forms GTP, glutamine triphosphate, which is immediately converted to ATP by the cell. We go along with several 4-carbon rearrangements where no energy is yielded. But over here, there are 2 steps. One uses 2 hydrogen to elevate a new carrier, FADH2. Now, we haven't heard of that yet. It's not NADH. It's a flavonoid, but it is going to act in the same way, almost, as an NADH, carrying high energy electrons that came from the acetyl group over here, eventually to go to the electron transport system. Following that, there's another step where a hydrogen is used to make NADH, and that ends up over here. So when we think about the balance for glucose in the citric acid cycle, its main point, we're going to evolve the CO2. We're going to make a net profit of 1 ATP per turn. So since there are 2 pyruvates coming from a glucose, you get 2 ATP from this system. You get 1, 2, 3 times 2 or 6 NADHs, and you get 1, 2 FADH2. So the point I'm trying to make here is 1 glucose makes 2 pyruvates and 2 acetyl-CoA. So you're going to produce 6 CO2s, and you're going to run this cycle around 2 turns. So we'll look at that total balance in just a moment. But we've just begun the harvest. You'll notice the harvest of ATP is still only 2 more ATP. Add that to glycolysis, and you've just got 4 as a net yield. But there's significant energy, mainly from the high energy electrons that came from the acetyl-CoA. They were transferred over here, and the hydrogen is tacked on, and these NADH and FADH2s are carrying a lot of energy. They're going to be transported to another set of enzymes, a co-adapted complex of enzymes that form the electron transport system. Before we do that, notice that in the text there are more complete drawings, and it's useful to notice the actual extent of our knowledge about this cycle. Here's the coenzyme A and the NADH and CO2 production. Here's the acetyl group, and notice it's 2 carbons and an oxygen with hydrogens on it. that is transferred in. Oxaloacetate, you'll notice, is a 4-carbon group, and the acetyl group basically sticks onto it. But it does it in a kind of a complex way. It's not just sticking onto the end. It's actually causing a rearrangement, because this is now, notice this carbon is attached to the central carbon. So we're binding and rearranging. It would be simpler for me to tell you that the acetyl group is stuck on, and then those 2 carbons are immediately bumped off the CO2, but it's just not the way it happens. The chemical rearrangement, the shape change from oxaloacetate to citrate, means that one of the carbons over here is the first CO2 to come off. But these are the steps. And by the way, every arrow, 1, 2, 3, 4, 5, 6, 7, 8 steps, has its own set of enzymes that are necessary and cofactors that are necessary to keep this cycle running. So it is here the citrate to isocitrate is just a rearrangement, and we make an NADH and kick off a CO2 as we make the 5-carbon form called alpha-ketoglutarate. The next step, alpha-ketoglutarate to succinyl-CoA, basically an NADH with coenzyme A to NADH and CO2, another one of these steps to produce succinyl-CoA. Here's that step from succinyl-CoA to succinate, where GTP is produced and immediately transfers its energy to an ATP. Here we have an FADH2 production, another energy yield in the form of electrons to those carriers, to fumarate. Fumarate to malate is just a rearrangement, consuming water, you'll notice, and NAD to NADH is produced as we return to the oxaloacetate. Now, this is the lowest energy form in the cycle. But in this form, it's ready to accept the next acetyl-CoA and run the cycle another turn. Now, I want to make a point about these cycles. These are carriers. So something comes in and is added to them. And then they basically change form. So it's not like it's moving around. What's actually happening is these forms are being passed from enzyme 1, 2, 3, 4, 5 around the circle to oxaloacetate ready in step 8 to accept another acetyl group. So you have to kind of understand that monomer-polymer arrangement and the rearrangements of stereochemistry in carbon molecules in order to understand these energy transfers. So from the citric acid cycle, we have yielded a number of high-energy electron carriers, NADH and FADH2, and in fact, you'll notice our total yield has only been 4 ATP so far. Somehow we're going to end up with 36. That means that the last step, the electron transport system, is going to account for the production of 32 of those ATP. So by far, the majority of the ATP production is being accomplished by electron transport. This is also called the respiratory chain because this is where we're going to use the oxygen. Now, these are pigmented molecules, so they were called cytochromes, which is cyto for cell, chrome for color. They are proteins. They're embedded in the inner membrane of the mitochondria to form what they called a pigment complex because the chemical nature of them is a pigment. They contain a metal ion. So iron is found in these pigments and also copper. So the concept for understanding energy transfer in the electron transport chain is sort of like a stairway with the donors being NADH and FADH2 coming along on the second floor and contributing their high energy electrons. to the first or the second electron acceptors. Now here's a difference. NADH contributes to FMN, flavonoid, and FADH2 contributes to a coenzyme Q. Those are different steps. And as a result, each NADH is actually going to account for the production of one more ATP. An NADH and its electrons will produce 3 ATPs as it transfers its electrons along, where the FADH2 will only produce 2. When it contributes, what happens is the hydrogen is kicked off as hydrogen ion, but you notice we used hydrogen before. So that's kind of a cyclical. Contribute the electrons, bind to the hydrogen, bring it over here, contribute the electrons to FMN, and kick the hydrogen off. There's no kind of net production or a loss of hydrogen. Same thing here. H2 is released when this contributes its electrons to coenzyme Q. So you can see those coming off here. Now, if you go back to the idea of a stairway, when you contribute the electrons from NADH from FMN to coenzyme Q, you transfer those. They release energy. Now, there's energy released as you contribute a pair of electrons to cytochrome B, cytochrome C, cytochrome A, and cytochrome A3, just like walking down a stairway. You're losing potential energy, and energy is being released. Now, along the way, although there are 1, 2, 3, 4, 5, 6 steps, there are only 3 of these steps where the energy is captured for ATP production. And I'll show you that later. The secret's going to be we're going to use the energy release to pump protons across the membrane and concentrate them on one side. It's then going to run through a special channel that's going to couple that movement through the protein channel with ATP production. So this stairway concept, you are losing energy, but I want to point out, when the 2 electrons get here, the cytochrome A3, those 2 electrons, it's like they're standing on the step. The 2 electrons at cytochrome A cannot step down until the electrons on cytochrome A3 are stripped off. And so this is where oxygen comes in. Why do we breathe? Why do we breathe so hard when we're using our muscles? Because we use so much ATP. And here's where the oxygen is active. Oxygen, and it's shown here as atomic oxygen, is accepting these 2 electrons and using 2 hydrogen ions from the matrix to form water. So here is where oxygen is used, the very last step. Think of it as the final electron acceptor from the electron transport chain and the production of one water molecule. Now, water molecule is a different kind of waste. In the citric acid cycle, we saw carbon dioxide. Now, that's a waste that you have to get rid of. So we're going to transport it through the blood to the lungs, and we're going to breathe it out. That's a true waste. But water, this is metabolic water. It's actually a product of these reactions. And so metabolic water here is actually a product that just goes into our water balance. So the production of water here, since it's our solvent, is just, it's almost, think of it as a water conservation step. It's 300 mils a day in a typical body. And it lessens the amount of water that we have to bring in by drinking. So this is our initial look at this electron transport chain. But we haven't dealt with the actual energy production, the ATP production. That was the whole point of this exercise. And here's what the inner membrane looks like. What we've drawn here is a very, very simplified version of a mitochondrion. Notice they've drawn, though, the phospholipid bilayer of the outer membrane. Here's the cytoplasm, where the pyruvate comes from. Here's the intermembrane space where the citric acid cycle will form. And here's the folded inner membrane. You can kind of see that over here, how it basically is surrounded by that intermembrane space. But the mitochondrial matrix inside definitely has two sides. What they've drawn here is one protein complex showing you the FMN, the coenzyme Q, then cytochrome B, C, A, and A3 in position to transfer the electrons along. Now, that point I made about ATP production, it basically rests on the fact that these carriers are going to pass these electrons along. So here we come on with one NADH to NAD, and when we transfer the FMN to coenzyme Q, it pumps two hydrogens across this membrane. That occurs right here. So when the FAD comes in and contributes it to coenzyme Q, it's already missed this proton pump. Now, that's a new use of the term. Hydrogen ion moving across the membrane, being driven by the energy of release. That's an active transport mechanism. And it's also a proton pump, which is because H plus is a proton. So we're pumping two hydrogens across here. Here go the electrons from Q to B, no hydrogen movement. That's part of just the energy loss that we talked about. On one of our first diagrams of this chapter, we showed that 40% was captured in the ATP and about 60% was lost as heat. Here's a good example. Q to B yields no proton pumping. But B to C, there is a proton pump. Here's two more hydrogens are moved across. Now, what does that mean? It means we're moving hydrogen against the concentration gradient. So this is definitely active transport. And it is enriching this intermembrane space with hydrogen ion. So the electrons are now on that middle step in the stairway C, and it goes to A. Notice there's no pumping there. So that's more loss. But when it goes to A3, it pumps two more hydrogen. So the point is this: two electrons from NADH is going to move 6 hydrogen ions to get to A3. From FADH2, it's only going to move 4. Now here's where the real doer, the protein called ATP synthase, is embedded in the inner membrane. And it is a channel for hydrogen ion. We've concentrated hydrogen ion over here by this active pumping. That's what the delivery of energy from eventually back to the acetyl group and the glucose. This is what its purpose was. So when you concentrate it out here, diffusion drives it through this channel. When 6 hydrogen move back through the ATP synthase, that's enough energy to put 3 phosphate groups on 3 ADPs and make 3 ATP. So if you look at the NADH alone, you pump 6 hydrogen and you make 3 ATP. If you look at the FADH2, you only pump 4 hydrogen, and that allows a net of 2 ATP over here. So this completes our picture. We have delivered electrons from these carriers. And if you look at a total glucose, you're going to deliver enough NADH and FADH2 to pump 6, double the number of hydrogens. So 16, hold on just a second, 64 hydrogen will be pumped to produce 32 ATP per glucose. Now here, the A3 is yielding its its electrons to oxygen and combining with some of this hydrogen ion to produce water. So what are we seeing? The oxygen reactant and the water product of our net equation is represented here. And as I said, by far the majority of the energy yield if you're just counting ATP. So this is the overall balance sheet for glucose conversion in cellular respiration. The glucose in the cytosol goes through glycolysis. Glycolysis means sugar splitting. It uses 2 and makes 4 ATP. So plus 4 minus 2, a net gain of 2 right here. But it also makes 2 NADH. Those NADH are transported here into the mitochondrion where they're going to be converted to ATP. But notice the difference here. I said you're going to be able to make 3 ATP per NADH, but here it's only showing 4. Why the difference? And the difference is right here. To get the NADH into the mitochondrion requires an ATP. So that is another minus 2. It shows up here on the balance sheet over here. These NADH from the glycolysis only produce 4 ATP. When we take the pyruvate, the acetyl-CoA, we're making, and these are numbers that are on a per glucose level. So 2 CO2 are made per 6 carbon glucose. 2 NADH are made. Coenzyme A's, we need 2 to make acetyl-CoA, and we're into the citric acid cycle. The citric acid cycle kicks out 4 CO2s. It makes 2 ATP from the GTP step, 2 FADH2s, and 6 more NADHs. So over here, we're seeing the delivery of 2 and 6 is 8 NADHs and 2 FADH2s to enter into the electron transport system. That yields at 3 per NADH, that's 24, and at 2 per FADH2, that's 4 more. And so you'll notice over here is our source and number generating 36 ATP. Now this is a kind of a balance sheet for one glucose being broken down completely. But it makes a point about the importance of the development of an enzyme system that can use oxygen and break pyruvate down further. To make a comparison, if you have an anaerobic bacteria, it can break down carbons, like if you look at glucose, it can break that to pyruvate, but the pyruvate is often waste. So the bacteria that break down sugars to make ethanol are a good example. You're basically throwing away that ethanol. You can't harvest it for energy. That represents a transfer of chemical energy back out of the cell. So if your net, just to make a simple point, if your cell is breaking down glucose anaerobically, it's getting roughly 2 to 4, maybe 6 in some cases with special additions, ATP per glucose. Whereas if you go over to an aerobic cell with mitochondria, every glucose is giving you 36 ATP. So even though you have more organelles, you have more complexity, you have more processes going on, you're getting somewhere between 18 times and, let's say 9 times the amount of ATP out of breaking down one glucose. Same food produces 9 to 18 times the ATP. And as a result, you can maintain these other structures, specialize your metabolism, you can grow faster. And on our level, on the whole body level, when we look outside the window today, what do we see? The plants and the animals that surround us. The ideal environments of our Earth, the terrestrial environments around us, and the shallow water in the oceans, where light and energy are abundant. In those most favorable habitats, what you find is an overwhelming abundance of eukaryotic cells with mitochondria, meaning that their growth and reproduction, when it's supported by aerobic respiration, displaces the presence of anaerobic bacteria, displaces anaerobic organisms, and basically dominates the landscape or the water. Now, there's other processes going on. Our body isn't just a slave to our nutrients. We've talked about glycolysis here for carbohydrate breakdown. We're going to basically make a couple points about carbohydrates, about proteins, and about fats now. But in addition to glycolysis of incoming nutrients, which we've just covered, there are processes of carbohydrate conversion going on in the metabolism of our cells. An overall process shown here in blue called gluconeogenesis, which means glucose production. Gluco for glucose, neo for new, and genesis for production. So our body has the ability to take these 3 carbon intermediates and use energy. So here's fructose bisphosphate back to fructose 6-phosphate, kicks a phosphate out, back to glucose 6-phosphate, back to glucose. So this is the way, if our glucose level drops low in, let's say, our post-absorptive state of digestion, we can, through burning of energy, convert it back to glucose and dump it back in the blood. If glucose is excess in the body in our absorptive or post-absorptive state, we can store that as glycogen, called glycogenesis. So here is the balance that actually explains maintaining our blood sugar level. And we see a couple of things that are actually very interesting. Here's the presence of a 3-carbon molecule called glycerol. We're going to see glycerol used later with fats to a great extent. It's glycerol that's going to produce a triglyceride. So we can produce that. We can use other carbohydrates and convert them. And by the way, when I say other carbohydrates, I mean maltose is broken down to 2 monomers. Lactose is broken down to 2 monomers. We can take those and convert them to glucose by going into this pathway. So it may be another 3-carbon intermediate. It may be the fructose 1,6 step. It may be the fructose 6 step. But once we get into this gluconeogenesis pathway, we can take it to glucose if we need to raise the blood sugar, or we can store it as glycogen if we need to lower it. So this really represents our carbon balance and shows the central role of glucose. If we're feeding on diverse carbon, and lactose is a great example, milk, sugar is a tremendous nutrient. Dairy is an entire food group in our nutrition pyramid or our nutrition scheme. And the reason lactose is so valuable is not because it's pure glucose. It's because we can take the glucose in it and the other monomer in it and make it into glucose. Sucrose, our table sugar, which is so popular because of its, it's basically behavior altering taste. Sucrose is 1 molecule of glucose and 1 molecule of fructose. So fructose from our table sugar comes in this box, basically converting either here or here on a pathway that will take it to glucose. So carbohydrates in general will be able to use this complementary pair of pathways, glycolysis to break down and gluconeogenesis to build up. Notice building up. Notice that if we're going to phosphoenolpyruvate through oxalo acetate, we're using ATP. So it basically, that glucose step is an energy burner. Now let's talk about lipids, because lipids, whoa, have they different from the beginning. Hydrophobic molecules that don't mix with water. In fact, phospholipids and glycolipids form amphipathic molecules. We use them to form phospholipids to form the barrier of our membranes. And we need special digestive processes to break them to micelles and chylomicrons and transport them through the lymph system in order to deliver them to the blood. By the time they go through and go around a while, we've processed them into mobile molecules called triglycerides. So how do we take the abundant energy in those bonds and convert them to ATP? Here, enzyme in the cytosol can convert glycerol, the carrier. By the way, glycerol is not a lipid, it's a carbohydrate, and it can be converted to pyruvate. Pyruvate can be put directly into the citric acid cycle. Now, the fatty acid chains that remain on triglycerides, I'll show you that in just a second, have to be broken down using acetyl-CoA in a new process. Look at this term, beta oxidation. And let me just comment on it with an overview first. Imagine you have a lipid chain that's 18 carbons long. And it's a saturated chain, so all of those side groups are pure hydrogen. So it's very compact, just a chain of 18 carbons, and it contains a lot of energy, but it's hydrophobic. Now we go back to what we learned about the TCA cycle, that what enters into the TCA cycle is an acetyl group, which is a 2-carbon group. Now that acetyl group, 2 carbons, how would that relate to a chain of 18? Well, 18 carbon chain would yield you 9 acetyl groups. So one of those fatty acid chains will run the Krebs cycle and the electron 9 times around, compared to the twice around for a glucose. And when it does that, it will make a huge number of NADH and FADH2, which are running the basic electron transport system. Now, that's 9 cycles per triglyceride, per fatty acid, but a triglyceride has 3 of those. So 9 times 3. A triglyceride is going to contribute a glycerol, which is 1 acetyl group, and 3 fatty acid chains, which is 27 acetyl groups. So think about this. 1 molecule, triglyceride, contributes 28 cycles of the citric acid cycle. So that's why it is such an energy delivery system. Here is the space-filling model of a triglyceride. The black and red end is a glycerol, and it's a carbohydrate. You'll notice that when we break this triglyceride into fatty acid chains on the left and glycerol, here glycerol, by the way, another feature of this triglyceride, it's a lipid. It goes right through the membrane. So it doesn't need transporter or special carrier right in, although there is some effect on getting it across that is dependent on insulin. So here's the glycerol. Pretty simple to explain that piece of the triglyceride. Goes to pyruvate making 2 ATP. Then goes into the mitochondrion and goes through the system as we see. This is one of this in the cytoplasm. This is one of those fatty acid chains. See the CH2s? And we have oxidized it in breaking it away from this glycerol. So this end carbon has a double bond OH. That's why we call it, by the way, a fatty acid. The COOH group that we see on this end makes it a carboxylic acid. Now let's look at what happens inside the mitochondria. These are absorbed into the mitochondria, 18 carbons. And the first thing we do is use an ATP, breaking it down twice. Notice we're knocking off 2 phosphate groups and adding in coenzyme A. So we're basically putting energy in to stick a coenzyme A on the end. The first acetyl group is shown here in a purple box. We then feed in another coenzyme A, but from the middle step to the bottom step, notice this, we make 1 NADH, a high energy product, and 1 FADH2, a high energy product. So if you look at this NADH, we're already in the mitochondria. It's going to make 3 ATP, and this is going to make 2. So we've used 1 ATP to do these 2 steps, but we've generated stuff that's going to make 5. So we've already made 4 ATP, but we're not done yet. Or we've already gained 4 ATP, but we're not done yet. Look at what happens here. We've broken this box off, and we've added a CoA to this box. So this has been reduced now to a 16 carbon chain. So we're basically going to go from right to left, eating these carbons off 2 at a time. That's how you get 9 acetyl-CoAs. Now this acetyl-CoA is going to start right here. And what we saw is the ability to produce 34 ATPs for each one of these. So you can kind of see how one chain is going to produce a lot of ATPs. And also it kind of relates to what we said about the digestive system, the hydrophobic fats and oils. Those are going to be harder to process. Those are going to be chemically difficult to stage. But once you stage them as triglyceride, you transport them around at the body. It's not quick because it's not soluble in water. But a steady supply of these triglycerides will run this system at a very steady rate. And that's why we say that at rest, it's mostly our fatty acids that are providing the ATP to place us at rest, maintain our alertness, and all of the metabolic processes that are essential. So the constant blood flow to our brain at rest. Fatty acids are providing that base. So when you take a single triglyceride, you are going to basically be producing many ATP from a single molecule. Beta oxidation is what's shown on the left. A lipid transport and utilization is tougher. You remember in the intestines here, we had to beat those droplets down to small form called micelles, which we transported in here. Now here, monoglycerides like glycerol and fatty acids are going to combine to make triglycerides plus other proteins, other lipids, and they're going to be released by exocytosis, absorbed by the lacteal, the lymph capillaries that are present in these cytoplasms. This is called a chylomicron. It is reduced in size, and the chylomicron is going to be transported through the lymph system and delivered to the blood. So it provides a really constant supply. I mean, think about it, whether we're eating high fat or not. If we're eating high fat, you're getting lots of lipid. But if you're not eating high fat, you're still eating cellular material, and cells are all wrapped in membranes. They all have a certain storage of lipid within them. But they also are utilizing lipid on a constant basis. The membranes themselves represent lipid in our digestion. So a constant supply, if we are in balance, we do use fatty acids to generate ATP within the mitochondria of the skeletal muscle. So a constant supply of fatty acids is healthy. If we have excess fatty acids, they reach certain points when those regulatory switches are thrown, we start sequestering those fatty acids as an oil within this vesicle. So it looks like oil, but since it's in a cell, it produces that tissue that we call fat. Adipocytes are really the only place where we put away lipid for future use. Lipids and lipoproteins are shown here, a pretty complex system. However, because of their hydrophobic nature, they just do not process well in water solutions like proteins and like carbohydrates. However, this is showing a different aspect of our lipid and lipid metabolism, that when these chylomicrons, when these micelles are transporting around, they reach the liver. The liver is processing them into different lipids. So this is very low density lipoprotein. Low density lipoproteins, the so-called LDL. Out here in peripheral cells, by the way, that means any cell in the body. We break LDLs down to release cholesterol. You know, we need cholesterol for all our membranes, for making certain hormones and other materials. And the cholesterol that's not used, if there is excess cholesterol in the bloodstream, it gets absorbed by high-density lipoproteins, the so-called HDL, that becomes a part of our lipid balance. Now, it was interesting. I first heard of cholesterol in diet because of its correlation with increases in heart disease. Cholesterol was the villain in the story. High cholesterol was bad. So people were encouraged to eat low cholesterol diets. And certainly anyone who's suffered from a cardiovascular problem is often asked to do that still today. However, we now recognize that these lipids are broken into different categories with different impacts, and therefore we have phrases in the common vernacular of the good cholesterol and bad cholesterol, and we on our dietary label are now distinguishing among those different categories. Not every cholesterol has the same effect, but one of the things we can definitely say is that because cholesterol has two elements. One, cholesterol comes in our diet, and we manufacture cholesterol as it comes in. That cholesterol has different roles in the body, one of them being to make membranes. You can't make cells, plasma membrane, organelle membrane. You have to have cholesterol to go along with the phospholipid. So it's not the universal problem that we see. It has some specific effects when it's present in unusual concentrations. Now, let's talk about proteins. Proteins are made of 20 different amino acids. The amino acids, only 20 of them, are in every life form, from bacteria to blue whale, from seaweed to sequoias, all of that stuff. 100,000 or 140,000 proteins are synthesized in the body. So they are the most diverse polymer. They also are the most abundant. If you look at the total weight of protein, it far outstrips lipids and carbohydrates and nucleic acids in our body. Each of these has a unique function. When we talked about those multi-step pathways, like thinking back to our detailed diagram of the Krebs tricarboxylic acid, citric acid cycle, all the same thing. We saw 8 enzymes. If one of those enzymes is mutant, then the citric acid cycle doesn't run. Basically, that fertilization does not result in a successful birth. So when we break down these amino acids, how much energy we get depends on the R group. Now, every amino acid has a central carbon and a carboxylic group, in addition to amino group, no carbon, and in addition to a hydrogen, no carbon. So if that R group is a hydrogen, you've got the amino acid glycine, and it only has 2 carbons. If on the other side end of the size range, you look at tyrosine, which I believe has 8, 9 carbons in addition to the 2, so 11 carbons, you could potentially get 5 acetyl groups out of that. So every amino acid is going to be broken down in a slightly different way, but the common element. What are we going to do with amino acids? We're going to make acetyl groups out of them. And the acetyl group starts. Now there's going to involve some processing with proteins. First thing you have to do with the amino acid is knock off the amine group. That produces NH2 in solution and is the reason for our urinary system, which is where we're going next. That takes energy. It takes some energy to process the protein and also some energy to get rid of the ammonia. But after that, the acetyl group in the citric acid cycle can actually yield energy to our overall metabolism. We have to break a bond that links the amino acids together called the peptide bonds, and we have to free those amino acids. Basically, the lesson is we're going to contribute them to the same pathways, the TCA and the electron transport chain, in order to get ATP. Now, 3 things contribute to a difference with protein, and this is one of the reasons for the popularity of the protein diet. But if you eat pure protein, you can get enough ATP, but your body is also working hard in order to process those amino acids in that way. Proteins are more difficult to break apart than the carbohydrates or the lipids. There's one repeating process to break down fatty acids. That's beta oxidation, and it yields a huge amount of energy. Whereas preparing a glycine, you get 1 acetyl group. First, you have to deaminate, then you have to spend energy to in order to get ATP out of it. So more difficult to break because of the more diverse nature of the side groups and their bonds. Number 2, a byproduct, ammonium ion. This is ammonia in water, and it's deadly toxic to cells, whereas our cells tolerate pH 5, pH 4, pH 3 pretty well. Our cells do not tolerate pH 8 or above. Our strict antimicrobial killers are high pH. So ammonia, sodium hydroxide, strong bases. When we want to break off an organic glaze, when we're cleaning our oven before those days of high temperature, when we just burn it off, the chemical things we use, oven cleaner, very, very strong base. So we're going to have a special system, the urinary system, just to deal with this protein byproduct. Number 3, proteins do form the most diverse and the most abundant polymer in the body. And so the protein metabolism, the proteins broken down to amino acids from our diet are not strictly available just for ATP production. We're making proteins on an ongoing basis, and we need the amino acids. We can only make 11 of them. 9 of them we have to get from our diet. So those proteins have 2 jobs if you think about energy conversion. Proteins are very diverse, and as a result, the specific chemical reactions are more abundant, more numerous, I should say. Amino acids. What happens when we have this deadly poison, ammonia? Well, we form ammonium in water. And basically the urea cycle taking place in the liver. When that ammonium arrives, it combines with carbon dioxide, another waste, to make this molecule, urea. Now, the 2 advantages of this. Urea concentrates the ammonium ion. Notice that now 1 molecule is carrying 2 NH2s. So it concentrates. This is very destructive to living cells. This is still toxic, but at much higher levels. So our body can tolerate the presence of this ammonia. And as it reaches a kind of a steady maximum concentration, we're going to actually shut off about 30% of the blood flow from the descending aorta and shove it through the kidneys, where this is going to be removed. So we have to have a constant ongoing production of urine for excretion in order to hold these concentrations low enough that our cells are happy. So going back to the overall idea of digestion, absorptive and post-absorptive states, the idea was we eat, we have an absorptive state which delivers those nutrients around the body, and a post-absorptive state when they've been delivered. But I want to kind of emphasize one more thing because of your future with diabetes. If you're going into health, you're going to be hearing about diabetes. I don't know a single situation in my medical history where diabetes would not have affected a medical intervention. We treat diabetics differently. And the reason is because of that hormone insulin. Now we see here a single kind of illusory cell and its relationships to this white area, the fluids around it. This is where the digestive system and cardiovascular system is dumping these nutrients. So up here, we look at what happens to blood glucose. Well, blood glucose is transported into the cell if insulin is present. If not, it is barred from entry into the cell. If insulin is present, what happens? This glucose can be stored as glycogen. It can go through glycolysis and break down for ATP. Insulin is needed inside the cell to store glucose as glycogen, so you don't have storage of carbohydrate without insulin. The gluconeogenesis then, because of the absence of glucose in the cell, is something you can't perform in the cell. So that's the carbohydrate pathway. Pyruvate can be used to synthesize amino acids. You see that over here. But its presence is cut down if you can't get glucose in. So you can see how it affects protein metabolism. So let's talk about proteins. Here's the blood delivering amino acids to the surface of the cell. Guess what? Insulin is needed to transport amino acids into the cell. So it denies you this second source of metabolism. If you think about it, if amino acids just went into the cell easily, that you could basically eat high protein and get around diabetes. But nope, you need insulin for that transport. Now, growth hormone is shown because growth hormone together with insulin actually tremendously accelerates the delivery of amino acids. And what are we talking about with this amino acid pool? Well, in the presence of different hormones, we've talked about growth hormone, sex hormones, estrogens and androgens, and insulin are necessary for various synthetic steps that result in our protein. So those diverse and abundant proteins we talked about, we can't make them as well without insulin. So that leaves us over here with fats and oils that are delivered to the membrane of the cell. If lipid levels are elevated, lipids, as you know, go right across the membrane, so you can still get fatty acids into the cell. That's why type 1 diabetics still do have a basal level of fatty acid in their cell. But most cells don't store fatty acid. And guess what? To take fatty acids and manufacture triglycerides, you need insulin. So you can get them in there, but you also need insulin to get fatty acids into mitochondria. So because of insulin's role in transport across membranes, the this is less, this is not a panacea. This is not a solution to diabetes. You still need the insulin. Insulin can basically convert fatty acids over to glycerol back and forth. Notice that insulin is involved in the transport, the acquisition, and even in some cases the polymerization of each of our major nutrients. That's why if your body stops making insulin because your immune system has destroyed the cells in the pancreas, the result of that is you obtain external insulin and shoot it into your body every day, or you die of a wasting disease. So absorptive and post-absorptive states are affected by a variety of things, and this figure is for your overall examination, introducing the kind of the main cast of characters, the big energy steps. So right here, we have lipid metabolism. Fatty acids can be, basically absorbed and utilized, but excess ones are released. They are then transported out to storage in adipocytes, even though most of our cells don't store fats, only the adipocytes. The body does not willingly let go of them. We do have the production of triglycerides to produce fatty acids and glycerol that can contribute down here to ATP production. Here we have the glucose-glycogen cycle. Here's glycogen releasing from storage down to glucose, to pyruvate, to support ATP production. And over here, this shows the protein pathway. We do have proteins in our body that can be broken down to amino acids. I think I made this comment, but if your body is kind of at a low nutrition state and needs more energy, and you have a fat reserve and you have proteins available, often the body will break down proteins to amino acids and feed them in at some point in order to make ATP and hold on to the fat that's stored elsewhere in the body. Amino acids can be released into the bloodstream by the liver. Protein breakdown in the liver does monitor the level of those amino acids. 11 we can make and 9 that have to be there in diet. So if cells are making a certain amino acid, but they're needed throughout the body, then that release and retransport occurs. When we look at our caloric expenditures, and again, this comes from taking a substance like, take sucrose, put a gram in a bomb calorimeter and burn it. It'll tell you how many calories are released when you burn all of that gram of glucose up. Doing that with lots of different substances in the physics lab gives us our estimate of calories directly correlated to their effect on our body. So here we are at rest, calories per hour under 100, slow walking 200, speed walking 400, climbing stairs, not only walking, but walking with a muscular exertion to raise our elevation. Constant activity like jogging, and finally something like competitive swimming. Competitive swimming is more energy expensive because in jogging, you're basically using your legs, your trunk, buttocks and back are involved, but the upper body is pretty much balancing and being carried along, whereas competitive swimming, you have the ability to use both sets of appendages and still the requirement to posture your body into what we call good form. So as a result, if you're swimming competitively and exerting yourself to the highest levels, the estimated expenditure of calories per hour is very great. Now, one of the things we do not list here are some of those ultimate expenditure sports. So we talk about jogging here, very high level for what amounts to a fairly enduring exercise. But if you talk about competing at the highest level with for example, marathon running in the Olympics, that's conducted at such a fast pace that its expenditure per hour would certainly go up and approach this range. We talk about the base metabolic rate, and we measure it based on our oxygen consumption. We always measure it under standard conditions, and this has a number of applications. We can suspect that there's some combined effect between the respiratory tract, the digestive tract, and muscle use. So we could measure base metabolic rate at a walking speed. For athletic conditioning, we can basically measure oxygen consumption, an important variable to know as we're reaching higher and higher levels of exertion. So somebody who's training to run, let's say, the 5K or the 10K on the Olympic track would be interested in the improvement of efficiency of oxygen use under heightened level of exertion. We have to remember the metabolic rate also affects another system, and that's thermoregulation. 60% of that energy was lost as heat. So we have to operate within a range of our environmental temperature. We all understand that if it's 110 degrees outside here and the humidity is 90%, the dew point is 85, we don't go out and do bicycle sprints all afternoon because of the thermal conditions. We also understand we have to wrap up in the winter. But it kind of focuses on how our body can respond within that range to regulate our temperature. That high temperature allows us to be active in all seasons, and heat exchange is basically controlled by physical properties. Conduction, your feet and fingers on the ground are actually transferring heat to the floor. Convection, as fluids like air or water move over your skin. Radiation is just the departure of heat from the surface of your skin as the heat of your skin is activating atmospheric molecules. And the only difference between radiation and convection is that here you're pushing those molecules along the surface of the skin, and here you're just letting passive heat transfer basically raise the energy. Evaporation actually comes from turning water to steam, which absorbs heat and draws it out of the body. Within that range of physical factors, we also have cardiovascular adaptations, and in certain places in the body, such as this appendage. This appendage is a place where the blood vessels are relatively close to the surface. But these blood vessels, the red arteries and the blue veins, can divert the main blood flow in response to the thermal balance. So on a hot day like this, the blood kind of comes in the artery in a core position, more toward the bone and the center of the artery, delivering blood to capillary beds up and down this appendage. In a hot environment, we would basically arrange the sphincter muscles on the capillary beds to divert the flow into these surface veins to return. So the hot blood from the heat release of metabolism is going to be flowing close to the surface, where it can basically flood the epidermis and use it as a big radiator, so that things like radiation, convection, and evaporation can dump heat at a maximum level. The core vein, this the brachial vein, which is deep, is relatively lacking blood during a warm environment. But in a cold environment, we're going to reverse that. We're going to bring the hot blood in, by the way, in the artery, in that core position, through the capillary beds, picking that heat up. But now we're going to deprive the surface veins of blood, and we're going to send it right up that brachial vein. And down here, the radial and ulnar veins are deep. That means the insulation. Here's where the blood is. Here's where the surface of the skin is. So it slows the heat loss because of the depth of the blood flow. But also notice something that's going on here. As we flow down here, we've lost heat all along the arm. And as we flow right here, this deep vein is also right next to the artery. So heat from the artery is heating this region. And as this cold blood is returning from the hand and wrist and forearm, it's being heated by the arterial blood, so that when it comes back into the trunk and enters the heart and goes to the lungs, those delicate tissues, there's not such a drop in temperature. So it's a kind of a countercurrent reheating mechanism. Shown here, warm blood from the trunk is transferring heat to warm blood returning to the trunk. So that completes the detailed metabolism, and thanks for sticking with it. I know this is physiology at its finest. Chapter 25, Metabolism.